Konnichiwa, watashi wa Adam desu.
Watashi wa Jordan desu.
My name is Ayana Isabe.
My name is Yoon Asmi.
And this is a presentation on the chemistry of nanoparticles.
First, what are nanoparticles in Japanese?
Nanoryushi.
Nanoparticles are any particles on a scale less than 100 nanometers, 1 times 10 to the minus 9. Sorry.
A human hair is generally about 100,000 nanometers, and nanoparticles can be made from a range of metals and non-metals, and sometimes have special properties.
This is an example of a nanoscale, and it goes from the smallest, which would be an atom, to the largest thing, which would be neurons and alveoli, which are in the lungs. We can see that up here, that's 100 nanometers, and anything less than that is where nanoparticles are. Our nanoparticles range from 110 to 14, which is around here. So it's around the size of viruses, bacteria, and that could be why the smaller nanoparticles get, the more dangerous they are, because the body treats them like a pathogen, so they can be quite dangerous.
Silver nanoparticles have antibiotic properties, so are used in socks, plasters, and deodorant. Titanium dioxide is hydrophilic, so you can coat windows with it, and it draws the water closer to the window, so it goes behind the dirt and automatically cleans the windows whenever it rains. They also have medicinal uses. You can create nano cages by coating a water molecule in gold nanoparticles and then drying it out, so you end up with a very small ball. You can then saturate it with a medicine, and it will safely take that medicine to any location in the body and can be triggered with either chemicals or light to release the chemical.
So since in gold nanoparticles, first, starting with Au3+, we added trisodium citrate, and heat it under reflex. The trisodium citrate bound to the Au3+, reducing it to Au0. It bound to the outside of the gold molecules and stabilized them.
Here is a video showing the process of synthesis of gold nanoparticles. Hang on, it's sped up about 24 times because the actual process is quite slow. So it goes black first, and then slowly goes red as the particles get larger.
These are the sizes of the reactants that we used, and as you can see, the smaller the amount of trisodium citrate used, the larger the nanoparticles, and they range from 14 to 110 nanometers. And the interesting thing about nanoparticles is that they have different optical properties when compared to the metal in bulk. For example, you'd expect gold to have a gold colour, but when it gets to the nanoscale, it ranges from kind of red and brick red colours. And the 110 nanometer gold particles that we used, when you looked at them, were brown, but if you held them up to the light, you can see through them and they turn purple. The reason that they have interesting optical properties is because light carries a magnetic field with it, which interferes with the electrons in the particles. That kind of repels them to the other side of the particle, and an oscillation occurs. The electrons move from side to side, and depending on which wavelength of light is used, they absorb different wavelengths of lights. So if they absorb it, then that colour isn't shown, so you get a range of different colours, and the one that is absorbed isn't shown, so that's why we get red.
UV spectrometry analyzes the wavelengths absorbed by the sample, allowing you to measure the absorbance spectrum of the nanoparticles. 日本語で言います。UV スペクトロメティというのは紫外加工分光分析機というもので、ナノ粒子に連続的に800ナノメートルから200ナノメートルの光を当てて、その金のナノ粒子が光をどのくらい吸収したかを調べる機械で、これを使うことによって、分光分析ができて、金のナノ粒子の正確な色、金のナノ粒子のばらつきの分布、そして溶液の濃度が分かります。今回は私たちはこれを使って、グラフというか、データを取りました。
This shows which different wavelengths were absorbed by different size nanoparticles. So these were the 4 sizes that we synthesized. And as you can see, the 14 nanometer, the red peak, absorbs about 519 nanometers. The green one, 28 nanometers, absorbs 527 nanometer wavelength. 50 nanometers absorbs 570 nanometers, but also the peak is broader, which shows it absorbs over a wider variety of wavelengths. And 110, the pink one, absorbs maximum at 580 nanometers, but also has a much wider wavelength, so absorbs a wider range of wavelengths.
This is an introduction to CB, which is called Cucurbituril. It's a molecule which is a polymer which ranges from a number of different repeat units. For example, up here we have CB5, 6, 7, and 8. So the more repeat units you have, the larger the diameter of the molecule. And because it's hollow, it can form a barrel with N representing the number of repeat units. So because it's a barrel and it's hollow, we can use it as a cage molecule. And so we can put things in it. And also it can be used to, we attach things inside it which attach to the nanoparticles, and they can coat the nanoparticles and stick them together.
So it actually bonds to the trisodium citrate attached to the outside of the nanoparticle, and that sticks them together.
日本語にすると、2回目の実験で私たちは CB5 というものを使いました。で、CB5 というのはこれが5つ輪っか状に連なったもので樽みたいな形をしています。で、これが金のナノ粒子と電気的な結合をして、加えることによって電気的な結合をしてどんどん金の粒子が集まって大きくなります。で、大きくなるとナノ粒子の大きさが変わるので色が変わります。で、どのぐらいその大きさが変わったかというのを私たちは調べました。
So yeah, on the second aim, our aim was to try and find the maximum and minimum number, number of CB molecules that we could add to a gold nanoparticle solution, and we wanted to try and slow down the reaction as much as possible without actually stopping it.
On the second day, I worked with Yun, and we thought we'd try to change the concentration of the CB solution in order to try and slow the aggregation of the nanoparticles to a minimum. We found that the lower the CB solution concentration, the slower the colour change. That's just because the lower the concentration, the fewer CB molecules there are in the solution. So fewer attached to the gold particles, so it's harder for them to attach to one another.
We did basically the same experiment, but instead of changing concentration, we changed the volume of CB that was added. We came to about the same conclusion. We had a ratio of 2,000 microliters of gold nanoparticles to 1.5 microliters of CB to be the best ratio. In this graph, the black line shows the UV spectrum of gold nanoparticles on their own. So you can see the peak where they absorb most. And then from the red peaks, are the initial, once we added the CB, and then over time it goes to the bluer peaks, and you can see that the amount of light absorbed at about just over 500 goes down, and the peak at probably 610 goes up. So you can see that the optical properties are changing.
So when we changed our concentration, we ran it through the UV spectrometer, and we found that, just as we expected, the optical properties of the nanoparticles changed. So we can see the black line, that represents the original one, before the reaction occurs. And then as the reaction goes on, it goes slower. And you can see that by the distance between the two lines gets shorter and shorter each time. And also you can see that it ranges from blue to a kind of, from red to a kind of greeny blue colour, and that represents the colour change in the CB.
So our experiment gave similar results, but as you can see, the distance between the lines is smaller, so our reaction went quicker. We really enjoyed our project. It was both fun and interesting. Probably the best part was that we weren't actually told what to do. We were given an aim, and we had to design the experiment ourselves to reach a conclusion. Our research is actually going to be used by a scientist in France who is currently researching nanoparticles.
Finally, we'd like to thank the organisers of the UK-Japan workshop, as well as everybody who's helped us throughout the project. Students and teachers, we're also very thankful for your warm welcome. And special thanks to Sam Jones and Setu for their help during our project. Thank you.
ありがとうございます。